A few moments later.
Hi, Selina. Hi.
Hi, I'm Kim, currently reporting live at UBCO 123 with breaking news. A student at UBCO was caught dead in the science lab located at room 326. The lead scientist investigating the crime scene, Lydia Lee, found DNA sample located around the body and on the weapon used. We have Lydia Lee on the line right now.
Thanks, Kim. We currently have 4 persons of interest that could have done the murder. We went to their office for further investigation. All suspects to give their DNA samples to prove they're innocent.
My name is David Jack. I'm an associate professor in the Department of Chemistry. On March 14th, I was in my office doing calculations on the blackboard and answering questions for students.
My name is Wolthers, and I'm an associate professor in the Department of Chemistry, and I teach enzymology. And on March 14th at 2 p.m., I was probably in my office reading papers or writing a paper, or I might have been in my lab too.
I'm Selina. I am the teaching assistant for BIOC and BIOC393 biochemistry lab. I teach 2 sections, and on March 14th at 2 p.m. I was in the lab with all of my students there, so many witnesses, I couldn't have done anything.
Hi, I'm Dr. Ed Neeland. I'm a chemistry professor, and on March the 14th, I was in the lab cleaning up.
With their DNA samples on hand, me and my teams were able to apply the concept of PCR, which stands for polymerase chain reaction, which can be seen on the following step.
PCR, or polymerase chain reaction, amplifies specific DNA sequences rapidly and with high sensitivity, allowing for the analysis of even trace amounts of DNA. In any PCR analysis, we first need to make a gel, which is created using 0.5 grams of agarose mixed with 50 ml of 1,X tris acetate EDTA buffer and SYBR safe. Electrophoresis is then run to separate the DNA fragments submerged in the EDTA buffer. The 3 steps of the PCR reaction is denaturation, annealing, and extension. Denaturation is the melting of double strands by heating the DNA to a high temperature of around 94 to 96 Celsius. Annealing occurs at around 45 to 65 Celsius, where complementary DNA strands will anneal to the template through hybridization. Extension occurs at 72 degrees Celsius, in which Taq polymerase will extend the primer along the template, making a strand that is complementary to the sequence of the template. The DNA ladder and each of the 5 samples were loaded into the wells. The ladder is crucial as it functions as a size reference point for precise estimation of fragment sizes during gel electrophoresis. Additionally, loading dye is incorporated into each sample to enhance density and facilitate visual tracking. The benefits of PCR is that it is relatively simple, fast, and cost-effective. However, it is susceptible to contamination, which can lead to false positive results. Additionally, it requires prior knowledge of the target DNA sequence, which is not always available, especially in forensic scenarios involving unknown samples. The 3 types of PCR analyses used in this experiment is RFLP, STR, and VNTR. The gels from all of these analyses will be captured using gel imaging system.
With these steps, we were able to narrow down the suspects to 1 person.
RFLP stands for restriction fragment length polymorphism, and they are created as a result of differences in nucleotide identity at particular positions in the genome that lead to loss or gain of a site for cleavage by restriction endonucleases, or RE.
Using the gel created for the RFLP analysis, the ladder that was used was HIN3 digest of phage lambda DNA, which was measured for the distance traveled by each band, while also determining the band size. These measurements were then plotted as seen in this image. The y-axis of this plot was set to the logarithmic scale and was used to yield an exponential trend line. The trend line was used to calculate the distance traveled by each DNA band from the DNA sample for each suspect. As seen in this table, it is clear that the blood DNA sample gathered from the crime scene perfectly matched the distance traveled by the DNA sample collected from Ms. Spence, or Suspect 3. Other suspects' DNA samples are further in distance to CS1 compared to Ms. Spence's DNA. This is an initial indication that Ms. Spence is the prime suspect.
STR or short tandem repeats analysis is used when dealing with limited quantities of DNA of uncertain quality, such as samples obtained from a single piece of hair, dried blood, or saliva.
The ladder used for STR analysis is a simulation TH01 allele ladder. It is used to distinguish between two suspects at a crime scene as shown in this figure. This figure is a depiction of the gel of TH01 genotypes of the two suspect samples after electrophoresis of PCR products corresponding to the TH01 locus. In this figure, the number of repeats in each allele is indicated to the left of the observed bands in the allele ladder. Using the gel created using the STR analysis, crime scene and 4 suspect DNAs were observed. As you can see, CS2 or the crime scene DNA is a heterozygote and has 6 repeats in TH01 on one chromosome and 9 repeats on the other. Therefore, CS2 has a 6-9 or 9-6 genotype at the TH01 locus. Lane 5 or suspect 3 also has a 6-9 or 9-6 genotype. It was found that suspect 3 has identical bands which represent the size of alleles and TH01 phenotype as crime scene 2 DNA as shown in table 2.
VNTR or variable number tandem repeats analysis is used to exclude the DNA of the investigator from the crime scene. This is done by extracting cheek cells from the VNTR locus. VNTRs occurs in anonymous regions, so they do not code for any proteins and are not linked to any phenotypic traits. However, VNTRs are often used to distinguish individuals using a DNA fingerprint. Since the D1S80 VNTR region can be repeated between 16 to 40 times, and the heterozygous condition is the most common, it is very unlikely that 2 people will inherit the same combination.
In the process of eliminating Ms. Lee as a suspect, VNTR analysis was employed. This analysis utilized the EDVO-Quick DNA ladder to determine the band size of fragments from Ms. Lee's DNA. Similar to RFLP analysis, the distance traveled by each fragment of the ladder was plotted against their known band size, referring to figure 2. The y-axis was again set to a logarithmic scale to generate an exponential trend line, which was then used to calculate the band size of the DNA fragment from the lane containing Ms. Lee's DNA. The band size is determined from the DNA samples of Ms. Lee's cheek cells, and the skin cells from the murder weapon were used to determine the allele size of each band. This estimation was crucial for determining the number of repeats for each allele and thereby establishing the D1S80 genotype for each DNA sample. As observed, the CS2 DNA sample exhibited a heterozygous genotype with 38 repeats in D1S80 on one chromosome and 14 repeats on the other. Therefore, suspect CS2 possesses a 3814 genotype at the D1S80 locus. On the contrary, Ms. Lee's DNA showed a homozygous genotype with 18 repeats. This indicates that Ms. Lee has an 18 genotype at the D1S80 locus. The allele size was determined using Table 1 allele sizes at the D1S80, which contained all the allele sizes of the number of repeats. This table is located in the lab protocol of Lab 7 and Lab 8.
Breaking news! The hospital just contacted us saying that the victim was actually alive. We have James, the victim, online right now.
Greetings, I'm James Chiu, an agent with the FBI. It has come to our attention that there has been a misconception regarding my involvement in a recent crime scene. Let me clarify that my presence at this scene was part of a strategic operation aimed at apprehending a serial killer. During the course of this operation, I had a significant encounter with the perpetrator. I made direct eye contact with an individual fitting the description of our suspect, a young woman with distinctive bangs, blonde hair, and appearing to be in her early 20s. This encounter has provided crucial insights into the identity of the perpetrator. Furthermore, DNA evidence collected from one of our suspects, Selina Spence, aligns with the description provided. As such, our investigation is honing in on Ms. Spence as the primary suspect in this case. I'm not dead, and we're after you, Ms. Spence.
You can hear the sirens ringing. The police are on their way to their house. Thank you so much, Lydia. Do you have any last words to say?
I just want to say thank you to all the BIOC393 lab TAs and professors for teaching us all this technique so we can solve this crime scene.
Thank you everyone for tuning in to UBC 123. Hope you guys have an amazing day.